Hi student, I am Dr. Sagar Kumar, MSc PhD. Now I am going to explain in the plant growth and development phases of the growth and growth rate. Both are very important for NEET exam. If you follow the entire thing, whatever I shown here, it is not necessary to be afraid about the NEET, any type of the bit, any type of the mathematical problem also. That I am assuring you. Now, first, this is the phases of the growth. Phases of the growth already he has given in the morphology, that root, that only for that only I drawn this one. And here in the NCERT book under plant growth and development, he has given one more diagram. Both the diagrams I am going to explain. From this also neat bits will be created. Now, the phases of the growth, phases means here the meristematic already we discussed. Meristematic means already cell dividing capacity cells are called meristematic. So meristematic cells is to present, and next will be the elongation. This will be elongation zone. He has clearly given in the morphology also. And maturation zone. This is the maturation zone. And in this chapter, he has given this diagram. Now we'll see what the sort of the characters each zone the cells used to have. This is also very important. Each and every point he has given, and he can ask in the NEET. For example, first meristematic cells, whenever you are seeing, these are characterized by some characters. They may given in the matching or the statements. Now they are the rich of protoplasm. Why? Because their protoplasm is highly active, because cell has to divide. All of you know that cell division you read. So actively whenever they are doing, the protoplasm will be the very rich of the protoplasm will be there in that one, and high dense, thick, because of the all the types of the nutrients, all the types of the requirements used to present in the cell for the cell division. So the protoplasm will be thick and high dense. And next, and rich of the protoplasm. Next, large nucleus. Comparatively, because here you remember one thing. These meristematic cells won't have the vacuole. Vacuole is the main characteristic of the plant cell, which is to occupy 80 to 90% and above also most of the cells. Such type of vacuole is not present. So the comparatively the nucleus size will be large. Nucleus is to apply for present. Cell wall is primary. What is primary? Primary cell wall means flexible, means whenever the cell is growing, the cell wall should not restrict its size. For that one, primary cell wall. Primary cell wall is made up of, in the cell you might have read already. Primary cell wall is made by the cellulosic fibrils along with hemicellulose and pectin. So such type of the cell wall is to present, and more plasmodesmata connections. Why are they? Plasmodesmata connections means from the cell to cell through the cell wall. Plasmodesmata is to present. He has given in the cell also. Through the plasmodesmata cell, so like the cytoplasm connection will be taken place cell to cell. So through that one, through the diffusion, the required nutrients and materials will be transported to the meristematic cells. So plasmodesmata connections means in the around the cell, so many plasmodesmata connections is to present to pass the required material from parent plant to the meristematic cells. So next is the elongation zone. This is the elongation zone, and A, B, C, D are the elongation zone. Elongation zone means here the cells of the meristematic cells used to divide And in the cell cycle, you might have studied, that is, from the G0 stage, it used to and the G1, from G1, G0 stage will be there. From the G0 stage, some cells, out of the two cells, one cell may be sent out as a permanent cell. So those permanent cells will come, will send proximal side, has written clearly. Proximal side means originating side. So these are the cells which are sending, permanent cells are sending towards the proximal side, and there will be vacuolation. Already I told that the meristematic cells are not having vacuole, but this vacuole used to form here. What is the size of the vacuole? More than 80%, most of the cells. So the cell size will be 8 times to 9 times, like that it is to increase. So these are the elongation zone. And after elongation zone, maturation zone he has given. Maturation zone means here that the cells which are the elongated cells, means cells used to reach maximum size in the elongation zone, and then they will travel through, it is to go inside, and followed by the differentiation. Differentiation means already I discussed yesterday also. The cells which are the permanent cells used to differentiate at epidermal cells, cortex, stele, endodermis, all the things used to form. And here one neat question is there. That is, elongation zone, if you take the transverse section here, a student has taken transverse section and he observed all the cells are alike. From which zone he has taken, he can ask. He has taken it from the elongation zone because no differentiation, all the cells are alike. That also, similarly, you can ask the questions. But if we take the transverse section here, he can observe, the student, epidermis, cortex, stele, and xylem, phloem, all the things he can observe. And externally, how you will find out? Is it possible to find out where the maturation zone is started? Yes, we can see where the root hair are forming started. From there, the differentiation is started. Remember, this is also very, very important point for the NEET. So, by seeing external appearance also, we can decide where it is started, differentiation is started. That is the best symptom, is the symbol is that formation of the root hair. And here he has given, this is the radical has came out and it is producing, and this is the elongation zone. Elongation zone, see, small thing will become big. From A, B, C, D up to here, it is the elongation zone. And here it is not elongating much, E and F. That is the thing. Next, here vacuolation and new wall depositions. New cell wall depositions means here the hemicellulose and pectin, what I told. Here it will be less, but here they will be more. That is the thing. And next, it will go to the maturation zone. In the maturation zone, wall thickening. And next will be the protoplasm modification. What is this? Now, wall thickening means a secondary cell wall may form, depend upon the cell. Parenchyma won't have the secondary cell wall, and collenchyma will have some corners, it is to have the secondary cell wall. But in the xylem, phloem, and whatever the fibers, trachea, all these things will have the secondary cell wall, very thick one, lignin is to present. And in the bark, it will be the suberin, and endodermis also suberin used to form. So such type of the secondary cell wall formation can be taken in the maturation zone by the wall thickening and protoplasm modification. What is this? Protoplasm is modified, means protoplasm is to modify without nucleus also. Phloem elements without nucleus, it is to present. So such type of modifications also can be taken in the maturation zone. But parenchyma and whatever the whatever the medulla inside the whatever is present, they are the not having the secondary cell wall. But in the secondary cell wall formation, which is to lead mainly in the vascular bundles, and next it will be the collenchyma, and it will be the in the complex tissue, some cells. So this is the explanation for this one. If you know this one, any type of the question in the NEET, this is the concept. You can give the answer. Now, see the growth rate. What is the rate? How much it is producing in the unit of the time. That is the rate in the physics, chemistry, and everything. Everywhere it is. Now you see growth rate per unit time, it has grown because it is under growth we are studying. So here mathematically we can see and calculate how much growth it has taken place in that particular organ or the entire organism. Now whenever you are seeing, it will be the two ways it will be measured mathematically. They are arithmetic and geometric. Arithmetic growth and geometric growth. What is arithmetic growth? Arithmetic growth means here he shown clearly. That is, you see, one meristematic cell used to divide and become two cells. One of the cell will become we send out from the G0 stage. Only one cell, one cell will be the functional, and it will divide, and again it will send. He has clearly shown by the arrow marks also. This is this cell, and the second cell, it is this one. Like that, it used to produce. So in each and every mitotic division, two cells will be formed. Out of the two cells, one cell will be sent out in the G0 stage. In which type of growth rate? We can ask. That is in the arithmetic growth. So whenever it is sending one by one cells, it means the starting after 10 divisions also the cell number of the meristematic is the same, but here the cell new cells are formed. So naturally, the thing that he has given in the graph also he has shown, that is, it is a linear graph will be shown. That is the height. If you take one by one, always one cells, it is to occur in the time, it is to increase the size. Generally, it is to happen in the root elongation. Understand? Now, the thing is, here each mitotic division gives the permanent cells root elongation. Okay. Now, this is here shown in the formula how to calculate arithmetic growth. The L means length of the time when we are taking that time, when we are taking the sample, that time is equal to length of L0. L0 means starting length, means the unit time before when the unit is started, and rate at the particular time. So here first length, final length, whatever is there, that is the LT is equal to starting length plus rate by the unit time. That those are the things. You can ask anything. And in that one, when you are calculating the rate will be the LT minus L0. Remember this. It is important. He has not given the book, but LT mathematically you are the maths. RT is equal to LT means end final length minus starting length. That's all. That is the rate in the unit time. Over. And now this can be shown here also, that I will discuss. Now, geometric. Geometric means here the each cell used to divide and forms two cells. Again, both the cells will divide and forms the cells. Again, 4 cells will divide and forms the 8 cells. 8 will become 16. But here One will give one will give one only permanent cell, but always the number of the meristematic cells are the same. In which type of the growth? You can ask, that is the arithmetic. In the geometric, the cell number is becoming double. Each type of the growth you can ask, that is the geometric growth. And here you should remember one thing. Unicellular organisms, note down. Unicellular organisms used to show geometric growth only because they are the unit cell, one cell only. All says one cell become two, two will become four, four will be either amoeba or the bacteria, whatever it will be there, but which are the unicellular. They also will come. This type of the questions also you can ask in the NEET. And it is shown in one formula, that is W1, that is the final size. That is the means always it is dividing. So size is it is increasing. How it is increasing? It may be the size, weight, number, or height. So many ways it is to show. So here W1 means final size is equal to W0 means initial size, e to the power of into e to the power of RT. What is e? Base of natural logarithms. All of you know in the mathematics it is to come. That is 2.7. That is the always standard figure like pi value. So here base of natural logarithm. So here it is whenever it is the growth is going on, the geometrical growth is going on, it is to become more and more. Each cell will be. Generally, it will present in the embryos in the starting, apical meristem and radical and plumule. They will have this type of geometric growth for some extent. Later on, it will be converted into the arithmetic growth. That is the thing, remember it. That he has shown in one diagram. What is this? This is the zygote. It has taken one division, become 2, and taken one more division, become 4, and it is becoming one more division, and it will become 8. Like that, it used to form up to here. It is a geometric. Now I shown this one. Later on, some of the cells here, that whatever he has given in the shown in the diagram also in the NCERT book, these cells will act in the terminal. What is the root apex or shoot apex? They will show the arithmetic growth. Means each cell you observe, it is producing one cell like this. Like this, it is coming out like this. These things, please observe it, because he can ask the question, which one, what is this one, different colors he may give, or he can ask, this is the tip, whatever it is showing, like that he can ask any question. And this, whatever the embryo of the zygote is dividing, it will come under the geometric. Now, this thing, whenever this geometric is going on like this, here he has shown one graph again here. This will be the sigmoid graph, means a shape like that it is to come. Once he has this log phase, the need, how it is? It is now you see here any graph is starting little bit away from there. It is starting. Why he may ask? Because the graph, it is the cell division. Initially some cells are there. Though are they dividing, so it won't be started from the zero. It will start with a little bit above. Those are the, what are this? You can ask. That is the initial cells or the single cell. So initially it is to go very slowly, because 1 will become 2, 2 will become 4, 4 will become 8, 8 will become 16, 16 will be 32, up to like that, up to 200, 300, okay. Then for 600, 1200, like that, and that is the very fastly it is to divide. That is called as exponential growth. The growth, whenever up to coming here, it will become very fast. Why? Because the number of the geometric cells become more, and how much time it will be continued as long as the cell division required material are available for that. Naturally, in the nature, it is not possible. After some extent, the requirement nutrients and other things is not available. So the stably it is to go. That is called a stationary phase. But exponential growth can be maintained. He used that word somewhere. Where? Getting of the foreign product from the biotechnology he has given. Getting of the foreign product for the recombinant DNA. There he used that one. Means there they used to add nutrients all the time. Means whenever it is using the nutrients, it will be sand always. That is the thing. So this is the stationary growth. And next, whenever you are seeing here, this used to geometric, it is given relatively, it is to efficiency index. It is given in the textbook. What is the efficiency index? Also email. That is also important for me. That adding of new plant material to the old one will be very fast. That is the efficiency index. Efficiency index will be more for the geometric growth than the arithmetic growth. That also you can ask. Now we'll see in this one, whenever we are seeing quantitative comparison, in the growth rate only quantitative. Quantitative comparison means how much quantity it is producing per unit time. Quantity is producing means here the some small leaf is there. After one day, it become this much size. So this is initial size is the 5 centimeters. It become 10 centimeters, square centimeters. At the same time, we taken one more leaf that is having the 50 centimeters initial size, and then after one day it will become the 55. Means the unit time is the one day. Just imagine it become 55. So whenever we are saying how much growth this leaf shown, that much that leaf shown means or the starting length, the final length by the minus initial length. That is the final length 10 minus 5, it will become 5 centimeters. Here 55 minus 55 centimeters. So here absolute growth is the same in the both. Absolute growth is the same because here we are not considering the initial which how much size the producer is having, means that leaf is having, that we are not considering. So absolute growth will be in the terms of this thing 5 centimeters. This one, this also the 5 square centimeters only. But relative growth means here relative means it is compared with something. What is that one? Initial size. Initial size means here this will be applied. This formula will be applied. It is not given, but please note it. It is what is this one? LT minus L0. Here we given arithmetic. So LT minus L1 means rate by L0 initial size. So here and 100 is percentage, relative percentage, how much it grown. For example, if you apply this one to this one, 55 final size and initial size is the 50, that is equal to 5, and by initial size is the 50. So 5 by 50 it will become 10. 10 divided by the 100, by 100 divided by the 10, it will become 10%. Remember, in the absolute growth, it is not possible to calculate percentage. In the statements he may give, absolute growth and relative growth both can be shown in that percentage. Wrong statement it is. Only relative growth only will be shown in the percentage. But absolute growth means it won't bother about the initial size. Only how much growth it came in the unit time. Now you see, whenever you are seeing all the things, it indicates. Now that you see, you follow the entire thing and read textbook. It is enough. You will get any type of question is touching in this type. All the types of the questions I discussed. If you think in this one, you can generate some more also. Please remember it. If you like this video, please give the like mark. And such type of the explanation given in the other videos also by me in the respiration, photosynthesis I done. Total up to now 25 videos I made, long videos. Information is very perfect information for the NEET. I hope you will see those also. If you want to see those things, please subscribe this channel and you will get all the things. Short videos means only NEET questions, how it will be given. Yesterday also I given one video. So such a way, what I observed, I am fairly telling you. Some people are doing, that is a digital board and wearing all the t-shirts and all the things. And if you observe it, what I am telling, how much quality it is. I observed it. It is very poor quality, not up to the mark for the NEET exam. Please remember it. You observe it. If you feel like that, then you subscribe my channel. NEET level, how to discuss, I discussed in all the chapters. And further also I am going to discuss like that. Please, if you realize that thing, subscribe my channel and see all the videos. Within 2.5 months, by the March, I will release all the videos. Okay, thank you.